Good morning, everyone. Hi. Can everyone hear me okay? Okay. I want to thank the organizers for inviting me, and I'm really excited to kick off the second morning of talks. So if you haven't met me yet, my name is Sohini Ramachandra, and I'm an associate professor at Brown University in ecology and evolution, and also I am the director of our Center for Computational Molecular Biology. And when I was asked to give a tutorial for CGSI this year, I wanted to use it, given the theme of this week, as kind of bringing together individuals studying population genetics and the microbiome, to think about how, for those of us who study data from natural populations or samples across, say, the human body, an initial step we all do in our analyses is a kind of data visualization, often a clustering approach to really understand what it is that we're seeing in our data. For better or worse, we might make strong inferences from those visualizations, but I think that process is something that we all have in common. And so I wanted to use that as a sort of jumping off point to think about applications of latent Dirichlet allocation and how they're used in genomics. And I'd love to hear from people who work on the microbiome, because I'm a population geneticist, about ways that LDA might be useful with those types of data. So this is not news to anyone in this room, but I think it is important for us to remember, especially any of us studying genetics, but those of us studying human genetics, that our problem is really keeping pace, as we saw yesterday, with the amount of data that's being generated, and trying to understand what patterns there exist, what signal there is in that data. Especially, it's amazing to me how I started graduate school as the draft sequence of the human genome was published. So in my, adult life, what I've seen is data sets suddenly grow to where even 1,000 individuals is no longer routine, right? Tens of thousands of individuals, hundreds of thousands now, if you work with something like the UK Biobank, is becoming really routine for us to look at. And even if we're just working with common variants, we are routinely looking at over 1 million sites assayed in a human genome. And so if we're looking at data like that, a natural first question for those of us who work in humans, but really in any species where we might be using genomic resources, is to try to understand to what extent a widely distributed sample might represent multiple subpopulations. And what's reinforcing those subpopulations? Is it barriers to gene flow that are physical? Is it barriers to gene flow that might be cultural? In the case of humans, are there niches that people are occupying that are, or organisms are occupying, that are defining barriers to gene flow between them? But we can also restate this, especially in this community, as a kind of computational problem. So I'm going to say that the question I want to talk about today is, how might we infer, visualize, and interpret clusters that are generated in population genomic data? So again, this is a common first step for probably all of us in this room when we get a data set, is to try to first identify clusters and population structure within it. And a lot of the work that we do in this is driven by a direct application of a model described by Pritchard, Stevens, and Donnelly in 2000. So as of today, Google Scholar says that this has been cited almost 23,000 times, combined with the independent development of latent Dirichlet allocation by David Bly, Ng, and Jordan in 2003. The total citations of this model are now approaching 45,000. And I'm not going to talk about PCA as a related way to think about clustering, but there's also been a lot of work on showing how non-model-based clustering approaches like PCA can give similar results. But I want to talk about model-based clustering today. So not only did this initial publication by Pritchard, Stevens, and Donnelly have a huge impact, but it also spurred a sort of cottage industry of model-based clustering approaches, some of which were driven by these authors, and involved modeling linkage, involved incorporating prior information about geographic location into analysis. The Pickrell and Pritchard paper, tree mix, sort of went a different direction and had kind of nested hierarchical priors to try to understand how related individuals are in sort of a genealogical way along with the population structure approach. There have been algorithmic speedups like variational Bayes in order to approximate the posterior distributions that people are trying to infer. There are also maximum likelihood approaches that move away from the MCMC of structure, and many other competing approaches. Olivier Francois has had a lot of work in this area as well, and people in this room, like John Novembre. But I think one thing that's really neat about the structure paper is to return to its introduction. It states very clearly what the mission is, I think, often when we have data from natural populations, just in the opening paragraphs. And so what the authors write is, in one scenario, the investigator begins with a sample of individuals and wants to say something about the properties of populations. In a second scenario, the investigator begins with a set of predefined populations and wishes to classify individuals of unknown origin. And this application, both of these applications, I think we see over and over again in our field. So let's just restate the objective one more time. The objective of this approach is to assign genomes membership into some latent clusters, which we're going to consider as ancestries. And the input is going to be a genetic data matrix and a tuning parameter K. The selection of K is an art unto itself. The number of ancestries, but this genotype matrix is familiar, I think, to all of us, but I'll just be pedantic for a moment. So it's going to be n by L, where n is our number of sampled individuals and L is our number of loci. And each entry is going to be a tuple for individual I. So we want to separate the two alleles because they might have different ancestral origins, and we might be interested in that. So here I'm thinking about diploid individuals. And in case anyone here hasn't seen the kind of visualizations people commonly make from structure, there are many ways to visualize output, but probably the most popular way is a stacked bar chart after a method that Noah Rosenberg initially did for a paper and then published separately as Distruct. So here's what output might commonly look like. So you might have sort of n individuals along the x-axis, and then these black vertical lines, which the investigator will put on the data or the output post hoc. And these will represent the groupings of the individuals that the investigator knows about a priori, but were not part of inference, right? So these might be things like, in a human case, language spoken or some cultural identifier or something like that. The y-axis will indicate, it'll range from 0 to 1, and it'll be this stack chart of membership coefficients. And I'll define Q again later. But suppose we are trying to look for 3 clusters in this data, then we might see a picture that looks something like this. So here, each individual is a vertical bar, right? And has membership mixed across these 3 clusters in varying proportions from 0 to 1. So what the kind of inference we often make from this sort of chart, right, is to say that the rightmost group has sort of homogeneous ancestry within itself, has maybe been isolated from the other 2 groups for some time. And whereas the other 2 groups have sort of similar ancestry, some level of admixture with these 3 latent clusters, share some common ancestry with this 3rd group. And for some reason, we have different labels for them in our data set, but their evolutionary history, we might think, is probably the same in some way, or not distinguishable based on this. So just to look at what people do in data, going back to those 2 goals stated by Pritchard et al. So we might want to take a data set and try to understand what sorts of ancestries lie in it. So here's an example from Andres Moreno Estrada and Chris Genoux and Carlos Bustamante's group, looking at differential population structure in indigenous Mexican groups throughout Mexico and samples that were taken at city-based clinics, which they called cosmopolitan samples, and then comparing the levels of both African and European ancestry and then putative Native American ancestry inferred from the indigenous groups between the indigenous and the cosmopolitan samples. So here they're using structure to show that the cosmopolitan samples have much more European ancestry than do their Native American samples, and are much more homogeneous across locations in cities throughout Mexico than are the Native Mexican groups as you increase values of K. Incidentally, in my various literature searches on applications of structure, this is the paper that increased K to the largest value, which was 24, which is quite interesting. So another example, the second goal that's stated by the authors is you may have some predefined set of populations and want to say something about individuals of unknown ancestry. And this is happening more and more often in population genetics with ancient DNA. And here's an example now from a few years ago by Pontus Skoglund and Matthias Jakobsen's group, looking at the ancestry of 4 ancient samples. So you'll see they're sort of wider rectangles so that you can look at them. So these are 3 Neolithic hunter-gatherers from Scandinavia and 1 Neolithic farmer. And the rest of the individuals are from publicly available data sets, like the Human Genome Diversity Panel and the 1,000 Genomes. So here we maybe have some existing notion of what extant human genetic diversity is, and now we want to see how would we describe these ancient genomes as a mixture of these ancestries that are still extant today. So this generative model that underlies structure is also used to process text corpora. And I think that can be a useful way to introduce the graphical model for those of you who haven't thought about what's under the hood in a while. So the way that you might think about assigning a document to latent topics, and then I'll talk about how to make the analogy to genomes, is that you would say, well, in this document, I observe a set of words, W. I observe n such words. This document is a bag of words. Right? So that's how text corpora are analyzed. And then there's a latent variable z, which is the topic for the nth word in the mth document. And there's also a word distribution for this topic, for each topic. Right? And then there's a topic distribution for each document. So we look at each document independently. And there are 2 Dirichlet prior parameters. One is the prior parameter on the per document topic distribution, and the other is on the per topic word distribution. Okay, so this is the graphical model if you're thinking about text. So for genomes, it's quite similar. So now I have genotype data, and so I'm going to think of a genome as a bag of genotypes across loci. And I have an asterisk here for genotypes because really I should have a second plate there that has to do with the 2 copies that I have, the 2 ancestries potentially that an allele, that a genotype's alleles can come from. Right, so now Z is the ancestry for the nth genotype. I guess that should be nth genotype in genome M. And there's an allele frequency distribution for cluster K at locus L. And we also have the ancestry distribution for that genome. Sorry about the typo. And these Dirichlet prior parameters. So here in practice, alpha is going to be the per individual admixture proportion in each of these M clusters. And beta is the probability, the prior on describing the distribution of a particular set of allele frequencies in population N at this locus. But in practice, there's going to be a uniform prior that's placed on these allele frequencies per cluster. OK, so when we interpret the output from topic models in genomic data, so given this genotype data and the number of latent clusters, we get 2 pieces of output. And the first is this Q matrix. That's what we always look at in genomics, right? It's the proportion of individualized genome that originates in cluster K. So it's an n by k matrix, and this is what we're used to seeing in these stacked bar charts. But we also get another piece of output, which is the P matrix, which is for each locus L, each allele's inferred frequency in each cluster K. Right? And again, there's this Dirichlet prior on how these membership coefficients are distributed, and in practice in structure, alpha has a uniform prior over a large range, and it's updated in MCMC using a Metropolis Hastings step. Okay. So I've been thinking for a long time about how in machine learning and topic modeling, when text is analyzed, the focus is on interpreting the P matrix. Right? What are the words that are driving the topics? If you were to analyze Yelp reviews in Los Angeles, right, you could probably find various topics that are based on location, ambiance, service, food, and that seems to be where sort of these text corpora papers are focusing. And in genomics, we just totally don't look at that at all. We look instead at what is the membership, but not what's driving the membership clusters. So first I want to talk about what we do in genomics and some work that my lab has done on just visualizing data, and I'll come back to the idea of P matrices. Okay, so suppose we're visualizing population structure in the 1,000 genomes using LDA. So many of us have done this. These are the locations of the 1,000 genomes in case you haven't used them. They come from 26 different sampling locations and are more than 1,000 genomes now. So what are the features that we see of human evolutionary history when we visualize this Q matrix? So here, this is at 225,000 SNPs. So here's what our initial k equals 2 plot might look like across these 26 vertical rectangles that represent the different populations. So here, probably most of you know that red is a proxy for African ancestry. These are the African populations on the left. These populations next to them that have mixed red and blue ancestry are US African Americans and African Caribbeans who are in the Barbados. So this represents the mixed ancestry these individuals have because of the history of slavery. There's also this level of red ancestry over on the right, which is the admixed Native American population, so potentially reflecting the African ancestry that exists in those lineages. And as we increase K, we see new clusters emerge. The order in which these clusters appear is related to how they would appear in a gene genealogy using genetic distances. If we look at FST between different regions, different geographic ranges, we could recapitulate this. But so here, as we get to K equals 4, right, we're observing a cluster that is associated with African ancestry, one associated with European ancestry, that's in blue, one with South Asian ancestry, one with East Asian ancestry. And as we keep increasing K, and as we arrive at, in this case, a value that admixture tells us is the best value of K, then we can see some interesting things. So we learn that that African ancestry in African Americans and Afro-Caribbeans is West African as opposed to East African, which again coincides with what we know about human history. We can identify ancestries for which we have no good extant proxy, so putative Native American ancestry, where here, maybe the Peruvians sampled in Lima are sort of the best proxy for what this ancestral genetic makeup might look like, but we don't necessarily need great extant individuals with this ancestry composition to potentially identify it. So there are multiple challenges in simply processing and visualizing these matrices that I think sometimes limit what we do as a community with LDA. So here's an example where I'm trying to be nice, but I think many of us can empathize with what was happening for the authors in the 1,000 Genomes Phase 3 paper. So this is their plot of results using admixture on their data. Okay. So how many of you have had the pain of having to try to take something like this and make it look pretty for a paper?
Do you mean by making the colors look nice?
Yes, by making it interpretable. Yeah. No one? Okay. I definitely have spent days of my life doing this, right? Yeah. Was that your PhD, John? Sorry. What were you saying?
Oh,
yes, good. Yeah, okay. So this is not, okay, good. All right. So you can think about the challenges of making it look pretty, as Eleazar said, in 3 different ways. Okay, so these are all, they sound trivial, but we all know the pain of trying to deal with each of them, right? And how it can really limit potentially what you might be doing with your data. Okay, so the first is label switching. This is the simplest of the problems. So you can imagine this graph that I already showed you, right? And that you just because of the process of the algorithm in a separate run of structure or admixture, you get a result that looks like this, where clearly there's a label switching thing happening. You should just have switched the colors of the 2 clusters in the second run, but the solution that has been arrived at for the Q matrix is the same, right? There's no difference. You just didn't assign the colors properly. Fine. Okay. So the next thing that can happen is what's called multimodality. And in practice, or what I call multimodality. So in practice, as we increase K, There can be multiple solutions that are supported under the data, and then if we keep increasing k, we'll start to see all of them, right? But we can get evidence of them much earlier in the process of increasing k than we might think. I think often nowadays people do sort of one run per value of k, especially if they're using maximum likelihood-based approaches. And you know, if you think about what you're seeing in LDA as recapitulating FSTs and things like that, especially if you're looking at fine scale structure, it might actually be quite interesting to instead do many runs and get a sense of how equivalent the various divergences you're seeing are. Okay, so multimodality, here's one example. So in analyzing the 1,000 genomes, if we do 10 runs at k equals 7, 5 of them converge on this solution. Right, but there are 3 of them where, so in this case, the new cluster is brown. So it's East African ancestry. Those are the Luya in Kenya. But 3 of the 10 runs actually differentiate Northern versus Southern European ancestry. Right? And the last problem comes in looking at many values of k, which you can think of as an alignment across k problem. So there's no simple way, well, there's no way by kind of inspection to align 2 matrices that have different dimensions in the columns, right? So there are different methods for post-processing your output from LDA. Typically, they work on a fixed value of k. So here I have 2 different runs, R and S. And actually, when I started thinking about this problem, because I was sad about how many days of my life I had spent trying to color these graphs, the existing approaches all compared full column permutations of one run to the other, which becomes really computationally costly as k increases. Right? But you might think about doing all these column permutations and then taking a distance between your 2 matrices, one permuted, one not, and identifying the minimum distance and saying this is the alignment of these 2 matrices. And then as you increase k, you just do the coloring by hand. So instead, what we decided to do is construct a weighted bipartite graph that is going to encode the similarity between clusters of these different runs. So we take a cluster-based approach to doing this alignment. And so the first objective is going to be finding the maximum weight bipartite perfect matching of clusters in R and S for a fixed value of k. And this is easily restated as just solving the assignment problem. So you have a set of clusters you want to align with another set of clusters of the same length. So here's an example of what this graph might look like within a mode. So here I have 2 runs, and I'm showing you each. So these are k equals 4. There are 4 clusters in 2 separate runs of admixture, and I'm showing you the best way to match them by doing cluster-to-cluster distance computation. So here, the edge thickness corresponds to Jacquard's index between the clusters. A kind of numerical Jacquard's index, and the edge opacity is the rank similarity from a cluster in the middle to a cluster on the left. Okay, so what's happening here is this mapping is just solving the label switching problem. So here, these 2 runs have arrived at the same solution, the same Q matrix, but the ordering of the clusters was different, and now by doing this cluster-based matching, we can color everything properly. But this approach also allows us to figure out when we might be seeing another mode, because now the perfect matching, the one-to-oneness between the sets of clusters is not so easy. So in this example, the 3rd run at k equals 4, actually, if we were going to match clusters based on the cluster they're most similar to, we would match both clusters 3 and 2 in the middle run to cluster 2 on the right. But in order to produce this one-to-oneness, we then go with the next best thing for cluster 3. So what does this correspond to in the data? This corresponds to identifying South Asian versus Native American ancestry in our plots. Okay. So doing this bipartite perfect matching actually lets you identify these modes quite easily without thresholds that you might need in the full permutation-based approaches. So the reason why I'm making this shameless plug for our implementation of this approach, which is called Pong, is because it was a really fun project, and it was led completely by undergraduates at Brown, who are wonderful, and who are now all software engineers who make more than their professors ever will, but that's good. That's progress. So it's available freely. It can be installed by pip. It's also available through my lab's GitHub site. So the first three undergraduates, Katie, Tyler, and Aaron, he started this as his thesis project, were the initial developers. And then I'll talk a little bit about work Elijah there on the right, who just graduated, has done in updating Pong. But the initial implementation and release were published in Bioinformatics. Okay. So here is an example, a screen capture of what Pong does. So this is after LDA has been run. So you've got all of your structure admixture runs. They're on your computer. All you need is a file that points to where they are with relative paths and the values of k. Okay. So now I'm just going to start the command line, and we'll see sort of what happens. OK, so here we have 100 or 80 matrices from LDA across k equals 2 to 8 for the 1,000 genomes that are being processed using fast algorithms to solve the assignment problem. So now all of the matching and the loading is done. You can open up your browser window and load Pong. And all of the coloring is done for you. Those buttons on the right will tell you about modes. If you mouse over, you get summaries of all of the ancestry distributions in the different clusters. Because this is in D3 in the front end, you can zoom in. You can highlight particular clusters if you want to see when a color is introduced, how does it change as k increases. If you go into the modals, then you can see not only the different solutions that you arrived at, but you can highlight differences among them to try to understand what were the ancestries where the colors were being sort of swapped in and out as for a particular value of k. You can download all of these in various forms quite easily to make your figures. So, OK, that was my shameless plug, but think of it as like undergrads are awesome. So, OK, so this was something we did that made our lives easier, but for a long time, what we really wanted to do, but Aaron went to work at Oracle and Katie went to Microsoft, was to look at the P matrix. So one idea is to say this kind of approach is applicable to LDA, which has many different applications. Why do we just look at genomic data? We could think about analyzing other kinds of data. But also in the realm of genomics, why is it that we ignore the P matrix? What could we be learning from it? What is being learned from it in the process of producing our output? Right? So if we think about these entries in the P matrix, we can think about PLK1 in the superscript as the probability of observing allele 1 at locus L, given that the allele originated in cluster K. And this is given to us, right? This inferred allele frequency. So we can directly ask the question, which loci have the most information for ancestry assignment? And for some reason, this is not something that people have been thinking a lot about in the last 10 years, although there are, of course, counterexamples. But there was a really nice set of papers about this in the early 2000s. And there are many, but one I'm going to talk about is by Noah Rosenberg, actually when he was in LA, when he started his postdoc at USC. And I think part of why the focus has moved away from this is because this was at a time where genotyping was still costly. And the identification of aims and the idea that chip design in order to assay diverse populations was going to be really important to do well. That was a thing for a while, right? From like, say, 2000 to 2005, and then genotyping just got very cheap, and people sort of didn't worry about that so much. But this was a really nice paper, and it also was very focused on microsatellite data, because that's what there was for the Human Genome Diversity panel then. So this takes an information theoretic approach at looking at P. Right? So if we think about the average frequency of allele J at locus L across K clusters, and I'm going to talk about this with respect to SNPs, because that's what we're all sort of using now, but you could do this with multi-allelic loci, and they actually have a lot more information per locus than a SNP does. So this is all generalizable. But what this paper focuses on is thinking about mutual information between Q, the ancestry, the membership coefficients, and J, the observed genotypes. Okay, so how do we think about how do we think about this information? So we want to think about it as the reduction in entropy of ancestry assignment once we have observed genotype J at locus L. So initially, if I have not observed any genotypes and I want to assign an individual membership, how do I do it? I have K clusters. What's the best guess that I can make? Random assignment, right? So what would that be? Right, so I could give them 1 over K proportion membership across K clusters, right? So when we do that, the entropy of Q is log K. So that is something that bounds the information that we can get in a locus, which is a challenge for interpreting the P matrix. But you can show quite easily that then knowledge of the genotype J at locus L is going to give you this form for mutual information between Q and J. So there really is a set of very informative loci, even when you prune for linkage disequilibrium, that drives the identification of these clusters. So this is now what I've been showing you from the beginning, 225,000 SNPs that are thinned for independence, because we should remember that's what structure and admixture, at least the vanilla models, assume. If I calculate mutual information on the P matrix that's outputted and take the top 0.5% of SNPs, so this is just over 1,100 SNPs, I get the exact same solution. Right? So there's very little, you can recapitulate this result with very little data, even when you're just looking at biallelic SNPs. So what are the most distinctive markers for a particular cluster K? So now suppose I want, K is going to increase, this is going to get more subtle. And I don't just want to know about mutual information, I actually want to know for a particular ancestry, are there very informative markers? So the way that you can think about this is to think about Kullback-Leibler divergence at a locus from cluster M to cluster K. So what is this? This is taking relative entropy between the allele frequency distribution in M and that of K, assuming the expected distribution came from K. And I know I'm showing kind of a lot quickly if you haven't thought about information theory, but you can see the form of entropy there in the numerator, this sort of minus P log P. And you see that in the top, right, I have P log P where the ancestry is in cluster K, and then I'm looking at what's my expected allele frequency, what's the entropy when I'm using the distribution observed in cluster M. So that's KL divergence if you aren't familiar with it. So now you can do this for all clusters M that are not K and take the minimum, and that is a kind of distinctiveness score, right? Okay, so if we do this at K equals 2, you get results that are not surprising on the one hand, but are also kind of intriguing, because sort of everything that you find in the top, say, 5%, if you rank loci by distinctiveness, are all enriched for GWAS results and selection scan results and kind of every gene you've read about if you think about differentiation between Africans and non-Africans. So the most distinctive SNPs are in the Duffy antigen receptor for chemokines, probably not surprising to anyone who's done a selection scan. You see cytochrome P450, multiple genes, multiple pigmentation genes, things associated with leukocytes. Elements that you wouldn't find in any one paper, but you find across studies of disease and selection. But as K increases, what's interesting to me, although I'm not going to probe into it today because these are kind of hot off the press and we're still trying to figure out how to understand them given that informativeness changes with, it's bounded by the log of K, so it's changing as K changes. But as K increases, you cannot predict these most distinctive SNPs just using FST. So there's something a little more subtle that's happening in the process of using LDA. Okay. So we've implemented distinctiveness now in Pong. You can visualize a sort of tree of how cluster memberships and distinctive terms are getting swapped as you move across values of K. There are various other little features like swapping in minor modes to the front end from the modal. But I want to point this out because there are various types of data sets that are using these approaches now where they, I think, will be quite informative. One is looking at genes that are driving tissue-specific expression patterns. This is from Matthew Stevens' group recently, analyzing GTEx data and using grade of membership models, sort of slight alterations to LDA, to cluster single-cell RNA-seq expression data. So here is just one part of a really nice figure that they produce where they're looking at clustering results in the brain, right, in different brain tissues, and they see difference between the cerebellum and the cerebellar hemisphere from the rest of the brain, which is quite interesting. But they also use this same idea of distinctiveness in order to identify the genes that are driving these particular clusters, which make a lot of biological sense. And this is a really nice paper with its own R package led by Kushal Day. Okay, so just to kind of end with some fun, part of where we started this project was one of the undergrads, Elijah, was interested in processing text corpora after he took machine learning. And so we decided that we wanted to look at some latent topics in a data set of funded NSF abstracts. So these are grants that were funded by the US National Science Foundation between 2012 and 2016. Words were stemmed in them so that things like biology and biological were not regarded as different terms and were reduced to biolog. Okay, so suppose we look at these NSF grants and try to identify two topics in them. If you had to identify the two topics that most kind of differentiate NSF grants, so what are the two cultures between like all of science and engineering in the United States? What do you think they might look like? I mean, not that you should have an a priori notion, but I don't know. What are the two disciplines that are most like rivalrous with each other? Yeah. I'm gonna get you ice cream later, sorry. Or give you the ice cream you're gonna get us. Right. So okay. So here on the left are the I think 10 or 9 most distinctive terms for one cluster. So you can see they have to do with sort of computation, maybe in the natural sciences also, some engineering, and biology, species, climate, plant, ocean, ecology, gene, genet and gene, different, water, evolutionary. Okay, so what does this look like if you look at sort of a Q matrix style plot? So it's quite variable across different grants. This is sort of bizarre to look at, but so you can see the third from the left is the Directorate of Biological Sciences. That's where you see most of those blue words that have to do with biology, also in geological sciences, and then just sort of everything else. The Social and Behavioral and Economics Directorate has a lot of variability between the two clusters. If you increase the order, it's individual abstracts. Oh, what's the ordering? It's sorted by red, like within. The ordering is just based on majority membership in cluster. It's a Pong thing. So it's not based on time or anything like that. But they were all funded. Is that?
No, just with that curve in the blue, there's some kind of sorting. But yeah, it's sorting on some average over the whole one.
No, it's sorting on membership in a particular cluster. I ran it for multiple values of k, so you can't see the bottom, but it's sorting on that. But in a given abstract, there's variability in membership in the topics, for sure. But yeah, it's kind of artificial sorting. Okay, so if you increase k again, you see something interesting. So sorry, this is kind of US-centric, but for those of you who write grants in the US and NSF grants, what are the two parts of an NSF grant? John, you said what?
There's the research statement and the broader.
So the broader impact. So how is your research going to help NSF fulfill its mission of improving science education for K through 12 individuals? So you can see that, so this is really, really important. But you can see that there's this Directorate for Education and Human Resources, which funds grants specifically looking at science education and pedagogy. And now this has kind of very solid membership in this red cluster. Okay, so now what's happening? So now what red is actually, what the terms that are most distinctive for red are completely terms that are used generally in the broader impact. So STEM education, student, conference, workshop. So I'm putting this up because I know you're going to work on the CGSI renewal in a couple of years. So
NIH
It's an NIH grant.
NIH analysis.
I'll work on it for next time.
Subcontracting from the grant.
Sure, we'll get working on the NIH ones. But for a career award, if anyone's writing one.
Also for the proposals that didn't get funded.
It's hard to get access to those. So you can scrape, right? But I would love to do that, and then actually you could just generate a grant and see what happens, right? That would be great. Except I'm not sure if a grant as a bag of words works so well, although, you know. And I think what's interesting about this cluster is that now the words learn and learning become distinctive for this cluster, and maybe that's why, one of the reasons why computer science has a lot of membership in this cluster. Okay, so on that note, I guess I just want to start us off, again, because I know clustering is an important way to look at data for all of us in population genetics and in microbiome research. So clustering is a common way to kind of generate your figure 1, but I think less attention has been paid to what is necessarily driving the identification of clusters. And now I think we're in a phase of really exciting papers and methods that are looking quite closely at this. So some recent efforts, John and Iran have been part of one, are using sort of PCA-based models to do selection scans. And this, I think, is a really interesting area. Alcus Price has a relatively new method out on this that shows that ADH1B is undergoing positive selection both in Europe and in Asia. And we actually see a signal of that using this distinctiveness approach as well. And there are, of course, many, I think, extensions to LDA that in the past were too computationally intensive to leverage, but you could imagine using it. I think Mark Salter Townsend and Simon Myers sort of did this with an HMM, a nested HMM recently, to do local ancestry deconvolution without reference panels. But you could imagine incorporating an HMM that samples ancestry sources and models linkage to try to do things like make population-specific recombination maps, like work that Daniel and John did, without having to worry about having great references. But it would be computationally quite intensive. And of course, it's important to think about challenges when we draw conclusions from LDA applied to genomics data, and John has written a great paper on this, and Daniel Falouche as well has put together a nice paper and a method called bad mixture that talks about how we can't necessarily infer past histories uniquely from what we see in LDA results. So with that, I'd just like to put up acknowledgments and again say that Elijah did a lot of the work on the distinctiveness, and I'm happy to take any questions. Everyone asked their questions already about grants, how to write them. Yeah. Mm-hmm. That's that Frobenius norm between the matrices as you do the matching. So it's generally quite high, right, in the 90s. You can threshold on it, so you can reduce how many modes you see based on the thresholding. We usually see it like above sort of 0.96, where 1 would be maximal similarity. But when it's slightly lower, it's because of the existence of modes. So, yeah. Yes?
So you've shown the sample size. Could you talk about the distribution of the sample size? Because some of the populations, for example, Africa and the energy from 3 different places.
Mm-hmm.
And one of the slides also showed that the West Africa ancestry
Mm-hmm.
and Ivory Coast ancestry.
Mm-hmm.
And I don't know if Ivory Coast is not part of West Africa. And how is the sample size distribution?
Yeah, so right. So sample size, like in PCA, is definitely going to affect the output from LDA, for sure. So were you talking about this slide? Just the. Sorry. Oh, the question was about how, I think about the 1,000 genomes and the sample size, the sampling distribution, and kind of how it is that we label some populations as belonging to certain geographic regions, particularly maybe in Africa, right? So this is for sure important to talk about, but this plot, or I don't think I listed sample sizes anywhere, but
if you go where we have the different ancestry before the predicate.
or the pretty, oh. Sorry, I'm not totally sure, but so I will say in this analysis, we kind of use, the Luya as a proxy for not West African ancestry, in part because they show this population structure that differentiates them from these other African populations. You're right that there are multiple Ivory Coast populations there, from Sierra Leone and from Ghana, so there are many problems with using the 1,000 genomes populations, especially single populations, as proxies for ancestries of individuals who no longer exist, and they may not be good proxies for individuals who were in those regions a while ago, and that's definitely going to affect your results here, and yeah, and that's going to be true for a PCA-like clustering as well, right? That's a good question. I mean, I guess. So so the question was, how do you leverage that? So sort of the lazy answer that's easy to give is to sort of say, well, if you think about things like LDA or clustering as data-driven approaches, right, they're going to be affected by the nature of your data. But I think one of the neat things about looking at the P matrix and taking some of the work on aims and moving it into sort of comparing what actual empirical data tells you about ancestry informative markers versus like inferred allele frequencies, I think it can help us understand a little bit about how FST and sample size might drive some of our identification of these distinctive markers. So there are some nice, like with information theory, you can end up with some nice ways of thinking about things very related to FST and sort of other known quantities. Yeah, Daniel.
So you were showing that the top snits driving the kind of clusters of the scene, they're all enriched for all those outliers we usually try to find.
Yes.
Usually if you try to find outliers, it's because we believe they're actually not representative of a neutral chemistry.
Yes.
So I was wondering if you were removing those snits, how would possible that could look like? Is it are we looking at something that is driven by the outliers that we're actually not really interested in prescribed infrastructure? Or would the problem or basically would the plot look very differently?
Yep.
If you were to look at
Yeah.
the lower kind of
Yes.
like, or is it just more messy because there's less information?
No, no. So mutual information is real. So two things I'll say about that. One thing I was trying to say is at k equals 2, you see like likely suspects, right? Usual suspects. As k increases, it's a little bit different. You see things that you wouldn't have necessarily predicted, some of which are unannotated, right? So who knows what it is, but then other sort of things that were identified in like validations of GWASs, kind of strange things. But yes, I should have had a picture in here. If you go to the bottom 50% in mutual information, you would just get uniform ancestry across these clusters in structure, because that's kind of the prior where you start, right? So you just can't move away from that. You really don't have information. Once you get to more than half of your data, then you start to get into some informative markers by chance. And admixture, which maybe makes sense also because of what it's doing in maximum likelihood space, will actually give you like just each individual will have complete ancestry, but in a different random cluster. So if you're looking at like k equals 4, like everyone will have 100% membership in a cluster, but it'll just be whatever cluster, right? So you do see you do see that there are a large set of loci that actually have no information. Yeah. which means their allele frequencies are just not significantly different across the ancestries. So, yeah. Okay.